Hello, everyone. This presentation will briefly talk about tobacco smoke, so a bit narrower in terms of chemicals compared with some of the presentations in the morning, but the idea is to show something like a case study on how indoor chemistry can be part of a broader research problem that involves not just chemistry, but other disciplines as well. So let me start by defining. We all know secondhand smoke. About 10 years ago, our research community started to differentiate between those direct exposures to fresh smoke, which we continue to call secondhand, from the multiple exposures that we can have, particularly in the home, where smoking may have taken place hours, days, or even up to months ago, and still, the chemicals are still there, and we may be exposed not just to the original chemicals, but to their reaction byproducts. So in that case, indoor surfaces play a critical role. When the smoke goes up, we see the cloud. A fraction of that goes away with ventilation, but an important fraction actually stays attached to surfaces, and we heard about that this morning, the important role of indoor surfaces as a medium where chemicals absorb, desorb, but also react. So I'm going to try to show some of the work we did in terms of those reactions. The exposures are not only inhalation in this case, there is a significant amount of exposure through dermal contact and even ingestion. If we think about, for example, small kids that tend to take things to their mouth very frequently, and certainly children are very vulnerable to this type of exposure for a number of reasons. So just to simplify a little further, rather than talking about the whole smoke, which contains hundreds or maybe thousands of chemicals, we will focus on just one chemical, which is nicotine. And there are good reasons for that. Nicotine is emitted by a cigarette in very large amounts, about milligram level per cigarette, compared with several orders of magnitude lower other organic chemicals. And also, nicotine is particularly efficient and absorbing to indoor materials and staying there for a long time. So it's an ideal precursor for indoor chemistry. So on the left here, we show reactions of adsorbed nicotine with nitrous acid, or HONO, as it's called in atmospheric chemistry. This is a compound that is produced in combustion, so it's mostly an indoor, primarily in this case, an indoor pollutant. It is also present outdoors, but it's higher indoors. The reaction is what we call nitrosation. Here is one example. The nicotine molecule reacts and forms this one here, which let me see if I can show. Okay, yeah, but you see the functional group over there, it's called nitrosamine. So there is a group of molecules that are formed that are all nitrosamines, and collectively we call them tobacco-specific nitrosamines, or TSNAs. We found 3 of them in surface materials exposed to HONO, first to nicotine and then to HONO in the laboratory. We use acronyms. Their actual names are very long, so we use these 3-letter acronyms. NNK and NNN are actually known to be present in fresh smoke. There is a lot of studies on them. We know they're carcinogenic, and we actually know their cancer potency. NNK is one of the most powerful carcinogens in tobacco, actually. So we found this chemical also being formed in these surface reactions. We also found chemicals like NNA, which is an isomer. It's structurally similar to NNK, and it's not present in tobacco smoke. So this is a unique, in a way, chemical to third-hand smoke. Our study could predict what would be the typical surface levels based on what we know from nicotine surface levels in a range that's about 300 to 300 nanogram per square meter. I will show you later some field measurements that confirm this. The second chemistry I want to mention today is the reactions of nicotine with ozone. We know ozone is present in outdoor air, is a part of urban smoke. It's brought indoors with ventilation at lowers that are typically lower than outdoor air, but still enough to drive much of the indoor chemistry that we know of. And so the reaction is essentially an oxidation, and I'll show on the next slide some of the oxidation byproducts from nicotine. These chemicals are going to be present, many of them, in secondary organic aerosols that are formed in this chemistry, and these are particles that have this sort of size distribution as shown here. A good part of them are ultrafine. That means they are to the left of that line below 100 nanometers, and these are particles that go deeper into the respiratory system. Now, don't worry too much about the details of the chemistry here. This is a chemistry symposium, so I had to show some molecules. But I mean, here's the take-home message here. The upper left there, that's nicotine. When it reacts with ozone, you look at the rest of the molecules, they have between 1 and 3 oxygen atoms on them. So we oxidize nicotine. And by doing that, these byproducts become more irritating to the respiratory system. How do we know that? Or how do we think that's the case? Because we can use a model that is called the Asthma Hazard Index that gives us a number for each of these structures. The number goes from 0 to 1, with 1 being the highest impact or the highest effect. And this is a model, so we had to take it with a little grain of salt. But for nicotine, this value is 0.64. All the red values in that table are higher than that. That means by converting nicotine to these other chemicals, we're actually potentially increasing their irritating potential. So by the time these are both the HONO study and the ozone studies, we have done that a few years ago. Around the same time, some colleagues in California were also interested in other aspects of what at that time was starting to be called third-hand smoke. So we decided to get together to create a consortium. The consortium is still ongoing. It's a really wonderful collaboration. And I'm mentioning here just the principal investigators from the 8 groups that are part of the consortium. These are from 5 organizations, mostly universities in California. This is supported by the University of California Tobacco-Related Disease Research Program. And we really could integrate very nicely very diverse research fields. As we see on the plot in the bottom there, this is our authorship map. So we can see that we really are making a big effort in working together, learning from each other's disciplines. And that's one of, I think, the principal reasons why I think this consortium is very successful. We show here the increase in the number of publications on third-hand smoke, which is mostly from the consortium, but increasingly also from other co-authors, some of which are here, Pete, where you are. I mean, we have some, I will mention later, growth beyond our consortium, which is really important. So the first thing we did was the first paper here, which is a roadmap. Where do we want this group of people, of this research to go? So followed by another similar study in which we not only update the roadmap, but also there is enough work done in 2017 that there is already a review article on everything that's been done on it. If you want to just read one paper, this is the one maybe to start with. So the next 3 or so slides, I'm going to give you an overview of work done by my colleagues in the consortium, in some of which we are involved, in some we are not, to give you, on one side, the idea of the diversity of the research, but also I will highlight how indoor chemistry informs biology or informs other disciplines different from what we do. One of the things we have been doing, particularly our colleagues in UC San Diego, they have been going out to various indoor environments over many years, taking a lot of measurements. The plot here shows surface nicotine. This is taken with wipes from various surfaces. This has been done in many, many homes, in cars, hotel rooms, and there is a study on a casino that is not included here. The main thing is that we found in non-smoking indoor environments, we did find nicotine in a majority of cases. So nicotine is a chemical that is found even when people are not actively smoking. For various reasons, we may bring it home with our clothing or being in contact with smoke outdoors or in other places. And some cases have to be with the history of the building. Certainly levels where there is active smoking, the surface concentration is 10 to 100 times higher or more. In casinos was actually much, much higher than that. The second thing on the table over there is another medium. Instead of the surfaces, we looked at dust. And this is a study from our colleagues in UCSF and UC Berkeley showing how, again, in smokers, but also in non-smokers' homes, we find nicotine and, in some cases, nitrosamines in the dust. Much lower levels in non-smoker homes, but the other interesting aspects here is in smoker homes, the ratio between the nitrosamines collectively and the nicotine is roughly what we had predicted in our study a few years earlier. Another thing, identifying good tracers in the environment and good biomarkers in biofluids is very important to quantify exposure. The ultimate goal here would be to differentiate exposure to third-hand with respect to second-hand smoke. So one strategy would be to look at this chemical, the NNA I mentioned earlier, that is formed in the surfaces but is not present in the smoke. So our colleagues in UCSF found these two stable metabolites from the reduction and the oxidation of NNA in animal models, in mice, at much higher levels than what we have typically in our system. So the current work is precisely looking at these biomarkers in human samples. So that's what's coming up next. The other approach is, rather than looking at this unique chemical, looking at a ratio of chemicals that have been used as biomarkers for a long time, but we may differentiate by looking at the ratio between exposure to fresh smoke versus aged smoke. And there is some good data here, both on house dust, that is on using, NNK and nicotine as biomarkers, or their metabolites, NNAL and cotinine as biomarkers instead of tracers. So, very briefly, a number of studies on the biology of third-hand smoke. At the top left here, we have in vitro studies looking at how, for example, NNA and NNK produce damage on the DNA molecule. We found that, in fact, those two chemicals have a similar effect. On the right, we see here effects measured in humans. This is our colleagues in UCSF have a chamber where one can age the smoke under controlled conditions and then deliver that to human subjects. And in this case, by inhalation, a couple of hours, what they found was gene expression associated with cellular response to the smoke. And that is a very short-term reaction. In mice, a number of studies, both in Riverside and LBNL, found effects due to chronic exposure. That's why animal studies, in this case, is necessary to look at exposures over the lifetime of the animals. And I don't have a lot of time to go through this, but there's a list here of effects that were observed, including carcinogenesis. 1 minute on this. What can we do? That's another aspect of what we are working on. What can be done about THS? There is some policy aspects. I'm not going to go too much into this. I'm just showing here a couple of state-level legislation related with third-hand smoke. In the case of a California bill, directly linking or mentioning our work. Other directions include public housing and consumer protection. Some of the work we are doing in our group using ozone, but in this case as a tool to remediate. So this is actually an industry out there using ozone generators, very high levels, of course, in an unoccupied space to remove not only tobacco smoke, but also fire and flood damage, among other problems. Okay, I won't go into the details here because I think I'm running out of time, but what I will say is that these curves here, the blue and the red, show the difference between applying these ozone generators in a clean chamber or in a chamber where we had smoked cigarettes 1 day earlier. So this is the stuff that's on the surface. A huge amount of ozone reacts with that stuff on the surface, and then we can see what's forming in the air, including these ultrafine particles. As we see here on the left axis, we see the particle size. So we did find the same ultrafine particles that we saw in the laboratory study. We see here in our chamber. And what's interesting here is that these particles run much longer than the ozone itself. So there is a question about what's the right re-entry time and what is also the long-term effects of using this technique. To finish, because I'm being kicked out here. As I mentioned earlier, I didn't mention earlier, but I should. Part of our consortium is focused on research translation. So that is making all this research available to a broader audience and engaging stakeholders that have a particular interest on it. And I will leave it at there. We have a really nice website, thirdhandsmoke.org, and this Resource Center is also very active in Twitter and other social media, and it's led by our colleague in San Diego State, Jörg Matt. Future directions, apart from the research itself, we have a research agenda there. There is some work that started to being funded by some federal agencies, HUD and NIH. I mentioned earlier, other colleagues are starting to work on third-hand smoke, and I think this is very encouraging. This is just a partial list of some of the universities in the US, Canada, Europe that are starting to be active on this. And in the case of the chemistry, indoor chemistry, some of the folks funded by the Sloan Foundation, right, including Pete DiCarlo, who's here in the audience. I don't know where you are, Pete, but yeah. And finally, one word about vaping. All what I described today is just conventional cigarettes, but of course now everybody is concerned about vaping. So there is a valid question, okay, is all of this, what I described here, these reactions of nicotine with all of these other atmospheric species, equally relevant when somebody is vaping instead of smoking inside their home? And I'm just showing here our study. We did measure emissions from e-cigarettes published recently in ESNT a couple of years ago. So thanks very much, and I'm sorry I'm a bit over time, just to show our colleagues at the LVNL who did much of this work, and our colleagues at the Consortium. So thanks again.
Thank you.